Our next speaker, Professor Jyotsna Batra. Ma'am has more than 10 years of research experience analyzing genetic variations for their association with complex human health disorders and dissecting their functional roles to identify new diagnostics and therapeutic modalities. During her PhD, awarded in 2010 in human genetics, she has published extensively demonstrating the positive association of alleles of several genetic variations with complex genetic disorders, 27 publications and 2 patent applications at the time of PhD. She has won 3 Best Poster prizes, 3 Travel Awards, and a Young Investigator Award from India's highly prestigious Society of Biochemists. Please welcome ma'am.
Thank you very much for the invitation, Yasha and Navneeta. Yeah. When I looked at the whole program of this lecture series, I was so thrilled. I thought, I want to come to this lecture series. You have really made it align with absolutely amazing speakers. And it was also amazing to see absolutely interesting questions coming up in the previous session. So I'm also quite ready. What I want to do with my talk is actually, if you have any burning question, just raise your hands, okay? You don't need to wait that I finish my talk. Yeah, I mean, there are new concepts, new ideas. May I actually know how many PhD students are here? Okay, pretty good. So this might be quite exciting, and the others are probably MTech students. That's what I understand. How many MTech students? Good, you've really separated yourself. That's also nice to see. So if you feel like there is any concept in my talk which you do not understand, because we all come from diverse areas, just raise your hands and I'll be happy to answer you. You don't need to wait for finishing like completely by the time that I finish my talk, okay? So as Yasha has already mentioned, that we did our PhD together at IGIB. And it's not actually just 10 years. I now carry more than 20 years of experience of working in genetics. When I did my PhD, I was doing genetics of asthma, and then I moved my research interest to prostate cancer. So before I go into the details of my talk, I also want to know how many of you have heard about prostate cancer? Okay, very few. And I shall explain the reason why you would not have heard about this disease, okay? Before I go into my talk, which is titled here, I'm going to talk about prostate cancer, a bit of GWAS. I'll try to give a bit of introduction to all of these so that, we can be on the same page. Genetic polymorphism will be another thing. And another word I would be touching in my talk is PSA, which is prostate-specific antigen. So, prostate cancer is actually the most commonly diagnosed cancer in men worldwide. When I'm talking about worldwide, it's mostly about developed countries. Yeah, I've been to a conference recently on reproductive biology where we have discussed a lot about prostate cancer. So it's a male-only organ, and this is why sometimes it doesn't come across so commonly, because it's a cancer which happens only in male, because only male organ. It is a male organ, basically. What happens in situation like India is that it's not as common, as I mentioned to you, that it's really common in the Western world. And it's very difficult to analyze the reason why it's not that common. The cancer of prostate is actually a disease of aging. It's most commonly diagnosed at the age of 70 years, sometimes at the 80 years. So for example, in Australia, 1 in 7 men will diagnosed with this disease by the time they are 75 years of age. While what happens in India, at least so far, the life expectancy was quite low, right? So and in case, men is the gender which actually would not go to the clinic until this is really a problem. So these are some of the inhibitory factors which has not made it to be as commonly known cancer in the developing countries, essentially. It doesn't mean that it's that uncommon. It's just that it's a disease of aging and less of the knowledge about this disease. So it's not just the most commonly identified cancer, but it also is the major cause of cancer death, especially if you have a cancer which comes early on. So there could be two types of cancer. One is the age-related cancer, and the other could be which is very early on diagnosed, which is around 45 to 50 years. And one of the most crucial part about this disease, which is prostate cancer, is that it's genetically and environmentally driven. So it's a combination of both the factors, like any other phenotype is. So this slide is not to show you that if you have a pet dog, you will start looking like a dog, or your dog shall look like you, but just to give you an implication that genetics and environment both have an impact on a particular phenotype. And as I mentioned, that prostate cancer seems to have almost 50% coming from its genetics. Now, I've been involved in what is called GWAS, or genome-wide association studies, where we try to look into the germline changes in human body and changes in the frequencies of what we call single nucleotide polymorphism. So our DNA is made of nucleotides, right? Of 3 billion nucleotides, there are some changes in these nucleotides which are associated with a particular phenotype. Of course, my study has been focused on prostate cancer, and I have been conducting the GWAS of prostate cancer. What do we do in GWAS is that we looked at genomic DNA, germline, so this is something which you are born with, inherited DNA, the frequencies of SNPs in a patient group, and compare their frequency in a healthy individual, the individuals who do not have the disease. And this analysis has so far mostly been done on a SNP chip basis. Now we can also do NGS. However, NGS is extremely expensive to do these kind of analysis, and we usually conduct these kind of analysis in a very big sample set. The current sample set which we have been analyzing, and this is a part of an international consortium where everybody has contributed samples to this consortium, and the acronym for that consortium is the Practical Consortium. That consortium currently has 400,000 prostate cancer patients recruited in it, and we have analyzed the genomic DNA of this huge sample set. Now, doing all of this analysis of looking at the SNPs, and which could be 500,000 SNP chip, this is the maximum we have run so far, but you can also then interpret rest of the genome, and that process is called imputation of the genome. And that imputation of the whole genome and looking at the differences between the two population cohorts, we have identified 400 such risk loci which are associated with prostate cancer risk. It says here 200 because we have recently published another Nature Genetics paper where we identified additional 187 risk loci, which is kind of making it to 400, and few other groups around the world have also contributed to this. Now, as I mentioned that each one of them, so if you have one SNP in your body which is really associated with prostate cancer risk, it would have very, very small impact. Like it increases 20% chances of having prostate cancer, or like twofold, yeah, something like this. But when you start to combine them together, and what is called polygenic risk scores, they have very impressive predictive probability of saying that this person have the possibility of having a disease in the future. I'm not sure if any of you have heard about the company 23andMe. It's currently a little bit in difficult situation with its consent form, etc. issues. However, the company already is taking genomic DNA from individuals. You just need to send your saliva and gives you a report saying that you have a probability of having a particular cancer up to this level. So these kind of companies, which we call as user defined companies, where user goes themselves, not by clinicians, they have been using our results and already commercializing the outcome of our genome-wide association studies. And it's also very important in those individuals where you do not know the family history of prostate cancer. As I mentioned, that it's a genetic disorder. So if you do not know the family history and you could just get your SNP chip test done, you would know what is like the familial contribution of the diseases. And that really changes how you should be monitored and how you should be given a therapy, etc. So that's the benefit of conducting GWAS, and it's like almost like 15 years we have when we started our first GWAS of prostate cancer. It came out in a big publication, and now we are really looking into doing it in multi-ethnic populations. So we have been able to conduct them in African American populations, also Chinese population and Japanese population. All of them have got their GWASs conducted for prostate cancer. There hasn't been a very significant GWAS yet coming out of India, unfortunately. And that's why I asked you, do you even know what prostate cancer is? Because there is lack of education about this particular cancer. So I've just put in up this slide here because I'm quite closely involved with the Indian Prostate Cancer Consortium as well. And we are trying to really make awareness to the oncologists as well as patients to become part of the effort of finding or helping us in research, which ultimately should also benefit patients. Now, once GWAS is conducted, your work does not finish there. I did mention to you the polygenic risk scores. That's like a direct clinical implication. But GWAS also tells you information about what genes nearby to those SNPs, which are associated with prostate cancer, could be used as biomarker or therapeutic target for the disease. And it's really kind of quite a mammoth task. Once you have conducted a GWAS, you have identified a SNP associated with the disease, which is based upon a huge region of linkage disequilibrium, right? That region from your genome inherit as such, as a component, yeah? And that's why we need to look into different ethnic populations as well. So we need to really find out which one of these whole linkage disequilibrium, which one of the SNP is really the causal SNP, which one is causing the disease, because the one which we have used as a marker could be tagging up to around 4,000 to 100,000 SNPs in that region. And then finding which gene actually it's impacting which of the genes or proteins functionality is getting impacted by this SNP is also an additional task. And we have been really doing it at a large scale, some of which is termed as fine mapping or post-GWAS analysis. And for today's talk, I will not have time to tell you about everything what we are doing, but just I'm going to take an example where we are much ahead in understanding where we stand in the post-GWAS era, which is actually an example from a protein coding chain. But I just also want to add that my lab is quite focused also working on microRNAs and long non-coding RNAs. And we are not just focused on protein coding genes, but are also looking at the other part of the genome. However, for today's talk, I'm just going to talk about one protein where we have done functionality or functional analysis in the post-GWAS space. Is there any question? If there was any concept not clear, I'm still happy to take question. Okay, I think that was the difficult part with respect to new concepts. The rest of it would be easier to follow because it's the classical part. So I mentioned to you another thing, which is PSA, which is prostate-specific antigen. As the name indicates, that prostate-specific antigen, so it's a very specific to prostate. It's something which is produced by prostate in a normal situation. It has a role in normal physiology in semen liquefaction. But what happens in the tumor situation, that the tumor starts to lose its architecture. And then the PSA starts to get lost from the tumor and enter into the bloodstream. And when it enters into the bloodstream, then it can serve as a very good biomarker. You can just measure its level in plasma samples, and which can give a very significant indication that a person could have tumor. Yes or no? It's not 100% specific, but it can give you some sort of an idea. Now, this is a very interesting biomarker, has been there for 30 decades. US and UK had used it as a screening method for the population screening. Then in 2015, they went to a stage where they decided that PSA test gives more harm than benefit and removed it from the screening. After removing the screening, 3 years after that, several studies came into play which actually indicated that the number of cases who were now getting identified with very advanced tumor have substantially increased, have increased up to 30%. So the discussion has now gone back that whether we shall implement the prostate-specific antigen test as a screening tool all the time. Australia never had it as a screening method, yeah? That's one very interesting thing, that it's a biomarker. And most of the world actually really think that PSA is a biomarker. But it also is a protein, right? And it's actually a protease, which has not really well established, but it should have some role, right? If the physiological role is in semen liquefaction, but in tumor also it shall have some role. So we actually wrote a review article, which is not just on PSA, but all the members of the same family, which is encoded by kallikrein, and I will show you a picture in the next slide, that how this could be very much involved in tumor remodeling at multiple steps, such as angiogenesis effects. It could have effects on the immune cell infiltration into the modification of the endothelial membrane, etc. So all the aspects of the tumor around, all the different parts where are involved, PSA could have a role, right? This is what we are anticipating based upon the other members, which are total of the 15 members of this family. All of them are actually work as protease, but they have different expression in different tissues, and their expression changes in different tumors as well. So from prostate cancer perspective, this locus here, do you see the pointer? Oh, yeah. This locus here, the 2, 3, and 4, and 5, this is most important for prostate cancer. It's androgen regulated locus as well. And always seem to be expressed or differentially expressed when you check tumor versus healthy individuals. Now, when we conducted our GWAS analysis, most of our SNPs which we have identified associated with prostate cancer risk were found to be in the non-coding part of the genome. Yeah, we never found any SNP which is present in the coding part of the genome. But the SNP in the PSA gene, and I would really like you to have a look at this, because I'm going to talk about this particular SNP, which is present in exon 4. This is the box here, over here, which if you could pay some attention to. So this is a SNP, and you don't need to remember this big name, RS176, whatever it says. But I want you to pay an attention because I said that it's first exonic SNP through GWAS, which we have found to be associated with prostate cancer risk. This 536 shows its transcript position. And because I will be using this terminology quite often, threonine 163. So this is the position in the protein sequence of the PSA gene where this SNP is present. So some individuals have isoleucine at this particular position, at 163 position. Other individuals will have threonine in this position. That's what this SNP actually causes a change. So it leads to a change in the amino acid. So it's a non-synonymous SNP. The other part I want you to pay attention to is the odds ratios, which is given here as 0.74, which actually tells you that frequency of this SNP is much lower in healthy individuals in comparison to prostate cancer patients. So if I want to just say it in a normal layman language, that the SNP could be protective against prostate cancer. And you could also look at p-value, and I also want you to look at this carefully because it's a norm to have just a p-value below 0.05 to be considered significant in our research. But when you do a population-based study where you have included, I think this study included 200,000 individuals, and this is what you could reach to the significance level. So we have excessive confidence in our results that this association is a true association. However, there was still a conundrum when we published this study first time in Nature Genetics, journal article. Clinician came and said, PSA is just a biomarker, right? It could not, if you have a SNP which is present in PSA gene, it's just coming out to be associated with prostate cancer because PSA is a biomarker for prostate cancer. And we are screening even our healthy individuals based upon this SNP. So this conundrum was raised, which we try to answer by multifaceted approaches, doing the studies in this population where there was no screening happening, and this association always stood its chance. It was always associated with prostate cancer risk. However, still could not clarify if it could have any kind of biological role. To understand its biological role, the simplest experiment And as I mentioned that, in fact, we have no clarity if PSA could also have a biological role, right? So what we did, the simplest experiment, is we took a cell line, which you would see here is a PC3 cell line, and overexpressed the PSA gene in these cell lines and wanted to check what is happening to the cell behavior of these cell lines. So these cell lines have no endogenous expression of PSA, even though it's a prostate cancer cell line driven from the bone metastasis of prostate cancer. So that's what you would be looking here as wild type PSA, the first graph here. Then you will see the terminology threonine 163 PSA, where we have done the site-directed mutagenesis to just do, so the same construct of a vector, but just did a single base change by using site-directed mutagenesis in these vectors, and then couple of positive controls, which we have to use, positive and negative controls. So as you can see in this graph, that by overexpressing PSA, our prostate cancer cell line start to show a better proliferative rate. They also started to show a higher migration rate. So proliferation and migration are normal phenomena associated with any tumor. Cells need to proliferate at a faster rate, and they need to migrate as well from one position to the other, so they can then make other nodes of tumor. So these are very important phenomena which we commonly check when we are looking for any carcinogen, essentially, or any oncogene which is involved in the disease. So overexpression of PSA caused an increase in cell proliferation as well as migration. However, we saw absolutely no effect when we have done the site-directed mutagenesis with the threonine variant. What you are looking here on the other side, MSK3, these are actually patient-derived organoids. So took the tumor out from the patients and convert them into kind of a 3D structure. So this is just a graph. In next few slides, I will also show you some figures. And we saw almost similar results, but much more deviation because they are organoids of different kind of cell lines. So we see a little bit of different effect. So that was our 2D culture experiment. Then we started to look at all these effects in 3D culture environment as well, which is considered like more closer to the tumor microenvironment. And we could also, by doing the 3D studies, like a spheroid formation with these cell lines, we could look at what really happens in the tumor environment. And this was actually a very surprising results, which we were not able to make sure it's because just changing 2D to 3D or they were the real impact. What you are looking here is change in the area of the spheroids, which we were making, and also the circularity. And both of these factors, when combined together, can give you an implication how aggressive tumor could be. And what you would observe here, that threonine variant, which we saw have no effect in cell proliferation and migration, was able to cause a very bigger sphere formation. So this was a little bit of surprise to us. And we observed almost similar impact when we again looked into the tumor organoid models as well. When we started to grow them in 3D culture, threonine variant seems to make very big spheroids and kind of started to grow as an aggressive tumor. So two opposite effects we have seen so far, but we're not clear if it was just due to 2D culture or 3D culture. So we thought, okay, let's really look at a co-culture kind of environment, as I mentioned to you, or probably not. Prostate cancer usually metastasizes to bones, yeah, and this is where it becomes highly aggressive because you could treat when it's in a local environment. When it's in a prostate, it's confined to prostate, you could use radiotherapy or prostatectomy to treat it. But once it has metastasized to bones, chemotherapy is kind of the only option left, and also is not as efficient as it should be because you have to treat the whole body and lots of side effects. So to create a better model, what we did actually, we used human osteoblasts and grow them in a matrix. So now we have really a bone-like environment, and then put our cells, which we have previously prepared, like PC3 cells, overexpressing variants of PSA, to see how do they really invade in the bone, essentially. And all these parameters combined together also showed us that the threonine variant overexpressing cell lines were invading very strongly into the bone microenvironment. So they were making all pseudopodias and migrating or invading at a much higher speed into the bone microenvironment. So this was all still in vitro culture. We then also wanted to see what happens in mice model. So we were able to implant our PC3 cell lines into the subcutaneous region of the mice to understand what really happens when we give it much more like environment which is more physiological, right? Like cell models are still where we are supplementing with serum and whatnot, all the components. So let's see what happens in mice. So in mice, when we implanted these cell lines, what we see is that with wild type PSA, tumor were much, much bigger in size within the same time frame in comparison to threonine variant. And that also goes with their tumor weight, etc. And after culling the mice, we have done several additional analysis with tissue cytometry, which Rupert has just shown you, which I haven't included in my talk here because I wanted to stay focused on the more important message is the second model where we wanted to check the metastasis. So I would just again quickly go back to this one to show you the tumor were much bigger with wild type PSA. But with metastasis model, where we have injected the cell lines into the mice heart so that they could then spread all over the body, the mice harboring threonine variant of PSA had really strong metastasis. It was so strong that within a very short time frame, we have to cull our mice. And the metastasis goes to liver, kidneys, legs, everywhere. So it was not just 2D, 3D differences, but what we could show in mice model that PSA actually seemed to have some sort of contextual role. So in the initial tumor growth, PSA was causing tumor promotion somehow, while for metastasis, the variant was causing the metastasis. And that gels with our spheroid formation data, which I have shown you previously. Now, if I go back to now patient samples, when I showed you the initial association analysis, I told you that it seems that PSA SNP is protective against prostate cancer. So having all of this complicated data together, we have to again then go back to patient sample and analyze what happens to these patients with respect to prostate cancer survival. And this was really kind of validating our data, showing that PSA have a dual or contextual role, because what now we see here is that individuals which have TT or CT or CC genotype, which we saw as protective, was associated with poor survival with prostate cancer, and higher mortality. So this is the data you are looking from is from 35,000 individuals where we could do this analysis. However, when we submitted this paper, reviewers still asked us that can you validate these results in additional patient cohort, and we don't see as good p-values here as we see in our bigger cohort because these are smaller sample size. So does not have that much confidence. However, it's the exact same trend that the genotype or allele, which was protective against prostate cancer, seemed to cause poor mortality also from prostate cancer. And all of our in vitro and in vivo data also speaks the same language. So then we try to also explain what could be the mechanism behind it. And one of the mechanism could be that, which is actually a previously known role of PSA, is as anti-angiogenic factor, which I also showed you in this big slide deck where the roles of other kallikreins were shown. So what we could see in this particular analysis, that the angiogenic effect of PSA was lost as soon as we have done this one base pair change basically to the PSA gene. And this is how probably it was removing the effect of PSA, which is in protecting from metastasis. So having more PSA is protecting you from metastasis. That's some of our results shows. We have also recently started to look into if the immune cells also play some sort of role. And this is a very, very early data where we have started to look into the patient samples and see how the genotype-wise distribution of immune cells are within the tumor tissue environment. And we are using also the co-expression of many markers, etc., using tissue gnostics technology. There are some preliminary data showing that there is a difference in the recruitment of CD45 cells, but we need to undertake this analysis in a much bigger sample size. Now, another very important aspect of the PSA, as I mentioned to you, is a protease enzyme. And it's used as biomarker, right? So what we wanted to see, what happens to its activity. Yeah, we have shown its impact, but what really happens to its activity? So with this one SNP change, what we could show that its activity is drastically reduced when we checked it against the peptide substrate, against the seminogelin, which is its physiological substrate, as well as several of the tumor markers, such as the data you are looking here is MMP2, which is very much involved in tumor metastasis. So the activity of PSA is reduced when you do just this one base pair change. Then we also do some molecular modeling of the PSA to understand what happens to its structure. And this data shows us that the kallikrein loop, which actually is quite important in recognizing its substrate, changes its dimensionality. It becomes much more flexible now. So there is a possibility that it's also changing the substrate it really identifies just by one base pair change in the gene itself. Now, again, going back to patient samples, so all of this work was again done by computational approaches, but very, very important and last piece of data which I'm going to share with you is what happens to this SNP and its association with the expression of PSA levels in human samples. So what we could see that this SNP was associated with much lower PSA level in mRNA at the tissue level, and most importantly, also in plasma level. So these patients, which have CT and CC genotype, inherently have low PSA level secreted in their plasma. And not just the free PSA, but there is also a norm nowadays, which I did not explain to you, to check the free to total PSA level in plasma, because PSA is not the best test. That also shown to be impacted by this particular SNP. So just to put all of these pieces and puzzle data together, this is the summary slide to give you the implication how our in vitro, in vivo, and then also patient-oriented data by doing all of this post-GWAS analysis of a single SNP have given us a huge indication that the individuals who could have this genotype would have inherently low PSA level, which means they could be protected against the prostate cancer. If they end up having prostate cancer, they will have highly aggressive prostate cancer. And it's already previously known that there are around 8 to 10% of prostate cancer patients which never has high PSA level in their blood. And these are the ones which have poor survival rate. They die within 5 years of getting detected with these diseases. So we are now currently at the stage of running clinical trials where alongside doing a PSA test, we are also checking for this SNP presence, and if these patients, these individuals have this SNP allele, which is the C allele present within their genome, we are recommending them to do a further follow-up with the additional PSA test, or also undergo the MRI testing, which is another imaging-based test before undergoing biopsy, or at least undergo much frequent follow-up exams. So this whole analysis, starting from multiple facets coming from really actually from patients, then we went back to the lab, and then really bringing it back to the patients with true clinical implications and verifying that GWAS has really given us lots and lots of important implications. And with that, I would really like to thank all of the individuals who are involved. It's a very big collaborative study. It has just got published in Nature Communications, and I could not give you all the details. If you're interested, you can go back and read the paper. First author is Dr. Sri Srinivasan, who was actually my first PhD student who started 15 years ago, and this whole work took her 15 years to get it published in Nature Communications. So it's really a perseverance, but such a beautiful implication clinically overall of the work. And now we're continuing with the clinical trial. So with that, I would be happy to take questions. Thank you very much.
Before we take questions, I'd like to compliment Jyotsna, Dr. Jyotsna, because I haven't seen a researcher of that eminence getting down to the level of a student and explaining right from what SNP is to taking to the level of what she's published in Nature. So I think my compliments to you on that. And I urge everybody to take full opportunity, because this is not easy to get Dr. Jyotsna to this platform, getting to interact with you one-on-one. So please,
I mean, if there are no more questions, I also want to just advertise our university Queensland University of Technology, which always is keen on taking PhD students at least. So we would be having our PhD program scholarship open call coming up soon. I think it opens in April with the submissions due in June, July. So I encourage you to have a look at it if you aspire to join PhD programs overseas. That would be something, and I would be happy to direct you as well towards that. Okay. Thank you then.
Now I request the convener of the event, Dr. Nandita Bharadwaj, to deliver the vote of thanks. Good afternoon. So as we conclude the second lecture of the lecture series, it's a privilege to express my heartfelt gratitude to everyone who contributed directly or indirectly in organizing the event. I thank our Honorable Vice Chancellor, Professor Prateek Sharma Sir, for gracing the occasion with his presence. I thank the esteemed speakers, Professor Jyotsna Batra and Dr. Rupert Ecker, for accepting our invitation. Your insights and expertise in the domain has left a lasting impression on everyone, and we are definitely looking forward for future collaborations. I want to thank the head, Department of Biotechnology, Professor Yesha Hasija, for the support and encouragement in organizing the event. Thanks are due to my faculty colleagues and their students for their cooperation in shaping today's event. I would like to thank Team PRO for recording the event. Thank you all of us for joining here and taking active participation. Thank you everyone.